So basically in diffusion of the, okay, you have to remember these, especially diffusion. Diffusion is a net movement of particles from a region of higher concentration to the region of lower concentration, but they usually won't ask this one. They would instead ask diffusion with water potential. Where did that go? Okay, anyway, that's diffusion. Okay, that's pretty easy. On osmosis, in terms of water potential, the net movement of water molecules from a region from higher water potential, which is a dilute solution, to a lower water potential, lower concentration. You know that like it's dilute solution because more water, less solute, more solute, less water, through a partially permeable membrane. High water area where there's lots of molecules, which is a dilute solution, or less solute. And water potential gradient, a difference in water potential between two areas. Gradient usually means a different. Osmosis is the diffusion of water molecules through a partially permeable cell. This is without the water potential terms thing. So yeah, that's all you need to learn. You need to only learn the definition and osmosis. Oh yeah, they had asked dialysing tubing in the P6 sample paper of term 1. The dialysing tubes, the holes are big enough for water molecules through, but not the sugar molecules. Dialysing tube is also called a partially permeable membrane because it lets some molecules through but not others. Okay, dilute sugar solution, concentrated sugar solution. It's just like all you need to learn is that the definitions are more than enough. Okay, and then osmosis in plant cells. Let's go for plant cells first because they have more weightage. Osmosis and plant cells. Plants do not burst in pure water. When plants are surrounded, this is a fully permeable, which means it'll let any molecules through. The pure more concentration solution inside cell walls and membrane. So then osmosis might happen. See, like for example here, although it's not easy, like blah blah blah, a cell plants in a pure water will take the in water by osmosis to its partially permeable cell membrane in the same way as animal cells. As the water go in, cytoplasm and vacuole will also swell. However, the plant cell has very strong cell wall around in it. This cell wall is stronger than the cell membrane and stops cell transfer from bursting. Cytoplasm presses out against the cell wall, but the wall resists and presses back the contents. So as the water, okay, this is what do you say? More concentrated solution is inside, and less pure water is outside pure water, and this is like concentration with solute, like more concentrated, and this is less concentrated. So osmosis, basically diffusion, but like in water, will happen. Okay, so what happens is the subvacuole and the cytoplasm will start to swell and become bigger. That's why in diagrams when you're drawing this, you have to show that the cytoplasm cell walls make bigger, like not too big, a little bump around these areas, and the cell wall will exert the same contact force, not contact force, that's in physics, but you know, get it. It pushes it inside. It's too strong. In plants, there are plenty of water in the states for cytoplasm. This pressure keeps switching, blah blah. This helps to support the plant. A plant state in this, like blown up titan form, is said to be turgid. The pressure of water pushing outwards on the cell is called the turgor pressure, meaning the here, right? You could see like the cytoplasm, the water will start pushing outwards. That's called the turgor pressure. Same, you just need to learn the definitions here. Flaccid, okay. Now imagine a cell wall in a solution more concentrated in than cytoplasm. Water will leave cell by osmosis. Cytoplasm and vacuole string, but the cell wall is strong and stays in position. The cytoplasm therefore pulls away from the cell wall. This pushes the cell wall membrane too, and then it looks like this. Here it's very important to show that this is disconnected from the cell wall. There's concentrated solution inside, concentration solute outside. Okay, this is called flaccid. Same, you just need to learn the definition and what it does. So basically like this part. Okay, and when the cell membrane tears from the cell wall, okay, when it completely tears out away, is called plasmosis. Plasmosis can kill the cell wall, plant cell, because cell membrane is damaged as it is tears away from the cell wall. So yeah, as well, just learn the definitions, it will be easy. Active transport. A cell needs these substances. Okay, then it has to do. Okay, so active transport basically means the movement of molecules or ions through a cell membrane from a region of lower concentration to a region of higher concentration against a concentration gradient from respiration. Okay, the energy. This process called active transport. In plants, root hair cells take in nitrate ions from the soil. The concentration of nitrate ions inside the cell root cell hair is usually higher than the concentration in the soil. The diffusion gradient for nitrate ions is out of the root hair and into the soil by the root hair active. Okay, so basically root hair cells take in nitrate ions. That's called active transport. Basic transportation. Carrier proteins or protein molecules in cell membranes that can use energy to change and shape, blah blah blah. Okay, there are special carrier proteins in the cell membrane of a root hair cell. The protein picks up nitrate ions from outside the cell and then changes the shape, blah, this entire thing. Just like learn the definition and examples and you're good to go. For example, diffusion is a result of particles moving. How does each particle move in randomly? Where does the energy for diffusion comes? Like that. Next, fourth chapter, biological molecules. Here, so in biological molecules, okay, first let's learn about there are three things you have to learn about: carbohydrates, fats, and proteins. Carbohydrates include basically, let's look at the definitions. Carbohydrates include sugar, cellulose, and starch. Like examples of carbohydrates are sugar, starch, and cellulose, but they contain carbon, hydrogen, oxygen. One molecule of carbohydrate is contains twice as many hydrogen atoms as carbon or oxygen atoms, meaning it's 6C6H12, so 6 times 2 is 12. That's what it means. The simplest type of carbohydrates with the smallest molecules are sugars. Sugars taste sweet, they are soluble in water. Same definition. Carbohydrates that have, these are the smallest, simplest carbohydrate molecules. Okay, one type of sugar. Okay, so wait, this is carbohydrates. From that you get like sugar. Okay, sugar is the smallest carbohydrate molecules, which is C6H12 and again O6. Okay, sugar, and from sugar you get glucose. Okay, glucose molecule is made up of 6 atoms, 12 hydrogen and 6 atoms. Its formula is C6H12, like the same thing. These atoms are arranged to form a hexagonal shape. It's a glucose molecules. Okay, this is a small starch molecule is made up from many glucose molecules linked up. You can and then they'll ask you this. You can detect the presence of starch using iodine solution. Okay, they'll ask how do you detect the presence of starch? Iodine solution. Oh, how is iodine solution used? Which glucose molecule are starch, something like that. Okay, from brown it changes to blue black if the starch is present. Most sugars can be detected using the Benedict solution. The Benedict solution is blue It changes to orange red when it's heated, okay, in a water bath with reducing sugars such as glucose. The color changes from blue to green and yellow. It's depending on the amount of reducing sugars it is. Okay, like that. Okay, this is a positive result, this is a negative result. And same, you just need to learn, not even learn these solutions, just learn reducing sugars at the most. Fats and oils are known as lipids. Fat is a lipid as solid, okay, and liquid oil is a lipid as liquid. Fats and oil contain 3: carbon, hydrogen and oxygen, okay. See, so then carbohydrates and fats and oils both contain, so what's the difference, okay? While carbohydrate is C6H12 and O6, okay, each molecule contains less oxygen that is found in a carbohydrate, okay? The oxygen is less 12 and then O and some, it's less basically. Fat molecules are made out of two smaller molecules, glycerol and fatty acids, okay. So this is fat, okay, made out of glycerol and fatty acids. How these join together to make a fat molecule? Oh yeah, it's like this. I mean, you have to remember that diagram, okay. And then fats are important for making cell membrane. They are energy stores. This is very important because this is a question. Okay, fat stores contain more energy per gram than carbohydrate stores, and suggest why birds that are about to fly build up lots of fats, because they are energy stores. Okay, and mammals often have a layer of cells containing fat droplets beneath the skin. The fat containing tissue is both an energy store and a heat insulating layer. So they might ask questions like this. Animals in thick layer in cold places, okay, suggest why. This is the answer. Okay, next. Fats and oils do not dissolve in water. They do dissolve in ethanol. So ethanol is used, okay. This tiny droplets of water is called the emulsion. So this is called the ethanol emulsion test, okay. This to find if there's fats and oils in the solution, like this. Okay, protein. Lastly, protein. Oh my gosh. Protein contains 4 elements: carbon, hydrogen, oxygen, and nitrogen. Some proteins also contain a small element of sulfur. A protein molecule is made up of a long chain of smaller molecular acids called amino acids. There are 20 different kinds of amino acids. Each amino acid is linked up together. Okay, if they are in a different order, they make a complete different thing, and there are infinite things. Proteins have different many functions. Their antibodies, hemoglobin, keratin are all part of proteins. Okay, they are examples of proteins. You just have to, I don't even think you have to remember this. To test for proteins, you use the Bio-Rad reagent. This is a blue and changes to purple when violet when contact with proteins. And this is like basically what you need to remember. Okay, so they might ask, where is that question? What is no, not this one. Where did that question go? Never mind. Yeah, that.